Good morning, and welcome to the inauguration of the new NIH seminar series, Mind the Gap. This year marks the 30th anniversary of the NIH Office of Medical Applications of Research and the Consensus Development Program. The program represents the recognition of the importance of translating NIH research into applications that directly benefit the public. Over the years, the process and methods of the Consensus Development Program have evolved along with the entire field of evidence-based medicine. Nevertheless, the core principles remain unchanged: the rigorous, independent, and unbiased evaluation of medical evidence and its implications for improving health outcomes. To date, there have been nearly 150 consensus conferences and State of the Science conferences sponsored by OMAR, the National Institutes of Health Institutes and Centers, and other federal public health agencies. In celebration of the program, we have initiated our Mind the Gap seminar series. This series will explore a wide range of issues at the intersection of research, evidence, and healthcare practice, particularly those areas in which conventional wisdom and intuition can lead us astray. The underlying intent of the series is to engage the NIH and health professional community beyond in thought-provoking discussions that challenge what we think we know and emphasize the importance of research in that process. It is particularly fitting that the inaugural speaker in this series is Dr. David Ransohoff, Professor of Medicine and Clinical Professor of Epidemiology and Director of the Clinical Research Curriculum at the University of North Carolina at Chapel Hill. David is a national leader in rigorous evaluation of research and its clinical application. He received his bachelor's degree at Harvard College, his MD at Case Western Reserve University, and his medical residency at Dartmouth. He completed the Robert Wood Johnson Clinical Scholar Program at Yale University and his gastroenterology fellowship at the University of Chicago. His long list of publications cover the spectrum of clinical studies in internal medicine and gastroenterology, as well as research methodology papers in emerging fields such as genomics, proteomics, and microarray analysis. To name just a few research areas that bear David's imprint, he has made important contributions to our understanding of the natural history of silent gallstones, cancer risk in ulcerative colitis, colorectal cancer screening and post-polypectomy surveillance, CT virtual colonography, and fecal testing with new molecular tools. In addition, he has written several methodology papers on the evaluation of diagnostic testing that I consider seminal contributions to the field. These papers on how principles of evidence can be applied to the myriad of diagnostic tests in development or already in common use truly serve as fingerposts for researchers, clinicians, and medical students. On a personal note, David is one of my favorite people to talk to and exchange ideas with. He's a true scholar and teacher with a broad range of knowledge and insights. I know that you will find his talk engaging. I won't steal his thunder and tell you why he's named his talk Chaos in the Brickyard, but you will see why the title is so apropos to this inaugural lecture. And you also see the scholarship, insight, and sparkle that have made David such a valued colleague to me. Just before I have David come to the podium, I want to recognize Kelly Marcial, Lisa Aramjan, Jennifer Crosswell, and Sue Rossi in OMAR. They conceptualized this seminar series and did all of the hard work behind the scenes to actualize it. I deeply appreciate their efforts in implementing the series and in so many other crucial activities of the Office of Medical Applications of Research. It's also a particular pleasure to welcome some of the previous leaders and participants in the Consensus Development Program, including John Ferguson, my predecessor as Director of the Office of Medical Applications of Research, and Bill Harlan, who was my predecessor as Director of the Office of Disease Prevention at the NIH. It's fitting that all of these people are here with us today for the kickoff of the seminar series that is a tribute to their contributions over the years since the inception of the program. In large measure, the success of the program rests on their important work and innovations. Finally, after Dr. Ransohoff's talk, and what I hope will be a lively discussion and exchange of ideas at the end of the talk, you're all invited to share some refreshments with us in the foyer outside of the meeting room. An added note by way of housekeeping, this seminar is being webcast. Also, when you have questions during the discussion period, please come to the microphones that are set up in the aisles. And now on to chaos in the Brickyard. David.
Barry, thank you for that nice introduction. I may ask for a copy of it to give to my mom and to my wife. It's very nice to be here on this occasion, the occasion of this seminar series, and also to talk about this particular topic, chaos in the Brickyard, translational research in 2007. Translational research is a very big topic, and the problems that I'm going to describe don't happen in all translational research. We can't hope to cover all of it. And we'll discuss which kinds of translational research. But problems can happen and be serious when different disciplines, it's translational, crossing disciplines, collaborate but do not understand each other's fields and the methodologic problems that can occur in each field. I've chosen the title Chaos in the Brickyard because Barry liked it when I talked with him about it earlier, and because of a wonderful essay that we're going to use that had that title written 50 years ago in Science by Bernard Forscher. This essay described many of the problems that we face in 2007, and I'm going to read about a third of the 750 words that he wrote, but I suggest you treat yourselves to reading this essay sometime. Listen to what he said. Once upon a time, among the activities and occupations of man, there was an activity called scientific research, and the performers of this activity were called scientists. In reality, however, these men were builders who constructed edifices called explanations or laws by assembling bricks called facts. When the bricks were sound and were assembled properly, the edifice was useful and durable and brought pleasure and sometimes reward to the builder. If the bricks were faulty or if they were assembled badly, the edifice would crumble, and this kind of disaster could be very dangerous to innocent users of the edifice as well as to the builder, who sometimes was destroyed by the collapse. And then Forscher describes how things changed. It came to pass that a misunderstanding spread among the brickmakers. The brickmakers became obsessed with the making of bricks. When reminded that the ultimate goal was edifices, not bricks, they replied that if enough bricks were available, the builders would be able to select what was necessary and still continue to construct edifices. It became difficult to complete a useful edifice because as soon as the foundations were discernible, they were buried under an avalanche of random bricks. Is this making any of us nervous? And saddest of all, sometimes no effort was made even to maintain the distinction between a pile of bricks and a true edifice. Now this essay identifies many important topics about bricks and edifices, about builders versus brickmakers, and about training. I'm going to focus on only one thing, bricks. If the bricks or facts were faulty, the edifice would crumble. We will talk mainly today about the strength of bricks or an individual study's results. Faulty bricks can be a particular problem when research is translational, type 1, applying lab discoveries to studies in humans, what we call bench-to-bedside research. We're not going to talk about type 2. It's important, but it's different, adopting practices in the community. If we ask, how do we know if a brick is faulty and could cause an edifice to crumble, the challenge in translational research is that it involves different disciplines, like in the lab, molecular biology or biochemistry, and clinical research that have rules of evidence to decide when a brick is strong that may differ. We're going to talk about those rules. So how do we know if a brick is faulty and might crumble? In a broad sense, we just apply fundamental principles of science that we are all familiar with. Like Richard Feynman discussed in this essay about science, pseudoscience, and learning how to not fool yourself. What is fundamental, Feynman said, is you must always ask yourself, what could be wrong with my results and conclusions? Feynman wrote, details that could throw doubt on your interpretation must be given if you know them. If you know anything at all wrong or possibly wrong, to explain it. What could be wrong? Is the brick or fact faulty? Could it crumble? This may sound simple to do and obvious, but it can be hard to know what could be wrong when the threats to validity or rules of evidence that we're going to discuss in one field like clinical research are foreign to those in another field like molecular biology or biochemistry. This fundamental principle, ask what could be wrong, was also the main point in John Platt's brilliant essay, Strong Inference. Platt illustrated that point by describing how in analyzing theories of antibody formation, Joshua Lederberg gives a list of 9 propositions subject to denial discussing which ones would be most vulnerable to experimental test. He was asking what could be wrong. Now, you may say this fundamental principle is nothing new, and you would be right. But Feynman was warning Caltech grad students, don't let yourselves be fooled. Platt wrote in his essay, of course this is nothing new, but people don't always do it. And the main message today is that asking what could be wrong can be particularly difficult when research is interdisciplinary, a message that fits with this seminar series theme of exploring issues at the intersection of research and evidence. Let's move to some specifics. I want to illustrate some specific problems in translational research in depth by looking at the field of markers for cancer, which is one that I've been directly involved in for a number of years. This is going to be just one example. Remember, translational research is very big, but it illustrates some larger lessons for other kinds of translational research. Molecular markers for cancer diagnosis and prognosis have recently received much attention in news reports describing disappointment, and I'm just going to show a few slides here to whet your appetite about what's been happening currently. Here's something from the New York Times. A new cancer test for serum proteomics for ovarian cancer stirs hope and then concern when results are not reproducible. In Nature, 2 years ago, a new proteomic test was heralded as the future of cancer diagnostics, but doubts about its effectiveness have begun to grow. In Science, getting the noise out of gene arrays. This isn't proteomics, it's gene expression. Thousands of papers have reported results obtained using gene arrays which track activity or expression of multiple genes, but are results reproducible. Now, there's been some improvement in the last few years, particularly in gene arrays, but there has been and still is in that field and others a huge inefficiency that we need to understand. Many weak bricks, piles of them. We need to understand why there is confusion and frustration, and in the future how to improve this situation. The point here is that if news stories about proteomics and genomics look kind of similar, it suggests that there may be some broad problems in how we are exploring omics fields and perhaps some other translational fields. With this as background, we're going to discuss markers for cancer in depth, where historically there have been many faulty bricks in the past, and we're going to start with some history of past claims about cancer markers, because history provides lessons, including lessons about what are threats to validity of clinical research. We'll discuss in the present current problems in detail RNA expression for cancer prognosis and serum proteomics for cancer diagnosis. And for the future, what are some lessons for conducting translational research to make it reliable, strong bricks, and efficient? Avoid piles of random bricks. Okay, let's start with history. What can we learn? The history of the validation of cancer markers is disappointing when initial claims are not reproducible. Non-invasive markers have been the holy grail of cancer diagnosis research for 3 decades, including carcinoembryonic antigen, a blood test for colon cancer, CA-125 for ovarian cancer, and magnetic resonance spectroscopy of plasma to diagnose any cancer. Do we in 2007 diagnose cancer by magnetic resonance spectroscopy of plasma? The answer is, of course, no. But does anyone here remember the study saying that we could? Raise your hand if you remember this. I'm serious. 2. Okay. All right. Well, it was published in the New England Journal and from a Harvard hospital. And it concluded that nuclear magnetic resonance spectroscopy is a potentially valuable approach to the detection of cancer. The NCI funded a study showing results were not reproducible. The study had a major bias. This article put the New England Journal in a refractory period for publishing articles about blood tests for cancer, according to people I've talked to at the New England Journal, a refractory period that lasted 20 years. Now, we might be amused that the New England Journal is in a refractory period about anything, but there's a lesson here. If our best institutions like Harvard and the New England Journal don't know when are the data strong enough to support claims about a blood test for cancer, then there is something genuinely difficult about the entire field about how to evaluate markers, what method should we use, and when can we trust them. Lessons were learned from the carcinoembryonic antigen experience. CEA was the blood test for colon cancer. After initial results in PNAS reported nearly 100% sensitivity and specificity for colon cancer, leading to high expectations, followed by disappointment, including a large multi-center study funded by the ACS and Canadian Cancer Societies, other studies showing much lower discrimination. The CEA experience led to development of the first rules of evidence to evaluate studies of diagnostic tests, a field established by my mentor, Alvin Feinstein, a clinical epidemiologist at Yale. And by the way, CEA research was, in its time, translational, involving immunology, read biochemistry or proteomics or genomics today, and clinical research methods. The rules of evidence that were developed from the CEA experience involved problems called spectrum of disease and specific biases that can occur in studying diagnostic tests. The field of methods and rules of evidence was very primitive then. This is a primitive article. And it's still underdeveloped, which is part of our problem in 2007. In contrast, the rules of evidence to study therapy like drugs, the rules there are much better developed. Now in 2007, cancer markers are even more promising than in the past because of new knowledge, like from molecular biology provides so many targets to measure. In the past, we knew little about what to target. Now we know the DNA path of mutations as tissue goes from normal to adenoma or precancer to cancer like colon cancer, and we now have assays that can measure targets. In the past, they were one-dimensional things. Carcinoembryonic antigen, fecal occult blood testing, PSA, prostate-specific antigen, they worked or they didn't. We've now got assays that are multidimensional. We can measure any target of DNA with probes and primers of protein with powerful mass spec technologies and other kinds of separation technologies. And so now cancer markers are very, very promising, but Mother Nature guards her secrets very closely. And new reductionist methods mean more data, but not necessarily more knowledge. The rules of evidence for understanding when an association is really there, when a brick is strong, have not changed. So our job is to explore new technologies and fields efficiently while avoiding predictable mistakes and inflated claims and expectations. We can't avoid unpredictable mistakes. Science is always tricky. But we should avoid the kinds of mistakes that after we've made them we say, gee, I should have known that. That's what's on the table today. And we need to make this exploration, this effort interdisciplinary and translational, involving molecular biology, biochemistry, and other basic fields, and clinical epidemiology and biostatistics, the fields of clinical research methods. But there is a culture clash that may hinder exploration when communication between fields is ineffective. This research is hard to design and interpret because of threats to validity. You can put people from all of these fields in the same room and try to have a discussion. You can put them on the same grant, but having them effectively communicate is a very, very serious challenge. And there's a culture clash when epidemiologists and biostatisticians are intimidated by the basic science, and the basic scientists don't realize that they've started to do epidemiology, even though they don't really want to, and they don't know what it is, but they're doing it, and it ends up being serious problems. Okay, we've done history. What about threats to validity? The meaning of the word validity is very, very broad. It just comes from the Latin word meaning strong, and it's so broad as to actually be confusing. There's probably 14 or more different uses of the word validity. We're not going to talk about them all. It's sort of interesting if you're a methodologist, but when you boil it down, there are two critical threats to validity. From chance. Does chance explain discrimination, for example, cancer versus not, or bias? Does bias explain discrimination? And we're going to look at both of these in some detail with examples. We're going to start with chance and an illustration from genomics. Using this paper that you've seen that describes how gene expression signatures predict survival in breast cancer, the lead article in the New England Journal, the front page of the New York Times, though it was below the fold. This slide shows a heat map in which tumor tissue at the time of surgery is examined for gene expression of 70 genes. Each column here is a gene. Each row is a patient showing expression of 70 genes, and the rows at the top are from patients with good prognosis signatures, below with poor prognosis signatures. So the gene expression of each patient, follow a row, is associated with prognosis, and the genes here are either down-regulated or up-regulated, depending on whether they're green or blue, so that down and up-regulation of genes is associated with outcome here, including development of metastasis, lymph node disease, and death. There is an enormous amount of information on this slide that, through the miracle of modern mathematics, can be reduced to a group with a good signature that has, at 10 years, an overall survival of 90%, while the poor signature group in red at 10 years has a survival of 50% for an absolute difference, 90 minus 50, of 40%. This degree of difference, this ability to predict prognosis, is better quantitatively than any other prognostic system like the NIH system or the St. Gallen system. And this strong discrimination led these results to be considered definitive for both clinical practice and biological research. For practice, gene expression patterns of primary tumors are better than available clinical pathological methods for determining the prognosis of individual patients, better than anything else that's available. And for biological research, the study that I just showed you provides compelling evidence that the genetic behavior of a cancer cell at the time of diagnosis defines its biologic behavior many years later, refuting a competing hypothesis that it's what happens after diagnosis that determines future behavior, like new mutations or selection by chemotherapy, and so on. The point is, if you believe the results of the study that I just showed you and that the results are quantitatively that big, I'm going to argue it's quantitatively less. Not that there's no discrimination, but that it's less. If you believe that discrimination is so strong, you might consider redirecting your practice if you're a clinician, but also what your research focus is if you're a biologist. But could chance explain results, or some of the results? The definition is that in multivariable predictive models, looking at multiple genes as predictors, overfitting, which is a problem of chance, can occur when a very large number of predictor variables, potential predictor variables like expressed genes, is fit to a small number of subjects. A model can be made to fit perfectly by chance, even if there's no real relationship. Rich Simon's group at the NCI showed how this works in a simulation in which they made up gene expression data on thousands of patients. It was all made up. And then they randomly assigned the label of cancer or normal to the patients, and then they did multivariable analysis to see if they could find discrimination. And they found that many commonly used methods of multivariable analysis produce a perfect fit, but it's by chance or overfitting. The basic problem is that if you let a computer run long enough in n-dimensional hyperspace, which is where it's running, and it's trying to fit an n-dimensional plane to your data points, it can find a fit, but the fit is idiosyncratic. The consequence is that the results of the model are not reproducible in an independent group. They don't describe reality or predict anything in the real world. This doesn't always happen, but you've got to check for it. And the method to check for it is to assess reproducibility in a group that was totally independent of the group that you derived the model from. So in the study that we just looked at in the New England Journal, can chance explain results? What might be wrong, as Feynman would say? This letter asked, in research to validate a prognostic system, the inclusion of 61 patients from the training group, that was published in Nature, into the validation group, this was what was in the New England Journal, means that the validation group, the New England Journal group, was not totally independent of the training group, and the degree of prognostic discrimination might have been inflated. And when the authors did a separate independent study, which they were doing before this letter was published, when the original authors did a separate study, this slide shows how much discrimination there was when different independent subjects were assessed. The same 70-gene assay was used, but survival at 10 years in the good signature group was 88%, in the poor signature group was 68%, for an absolute risk difference of 20%, not 40% as in the New England Journal. There's discrimination, it's just not as great. Now, it's not totally clear that overfitting is the entire explanation for this difference. There may be other explanations that can't be sorted out as well, so there's a differential diagnosis here. But in any event, if the degree of discrimination is less, would the interpretation that we saw before be so strong? This is what we saw before. My hunch is no. The strength of this interpretation depends in part on the degree of discrimination that gene arrays are supposed to give us. This slide shows how to check for overfitting by assessing reproducibility in a totally independent group. You start with an initial group of persons with cancer in blue and without here. You split them into a training set that's above this line, used to derive a discriminatory pattern or specific analytes. This is discovery. And then you apply what this pattern or analytes was to the subjects that had been split off this validation set to see whether the pattern discriminates in subjects independent of those used in discovery. Overfitting occurs so easily here in training that until you've tested the hypothesis you've generated here in a totally independent group, you can't make any claim that it works. Here's an example of where chance is successfully successfully addressed in a study of RNA expression. Again, multi-gene assay to predict recurrence in breast cancer, because the method showed independent validation was done when the authors wrote, the prospectively defined assay methods and endpoints were finalized in a protocol on this date, PCR was done on this date, and the results were transferred to the blinded data analysts for analysis on this date. We know that this was independent validation. What happens in some other studies, many people think, is when authors don't get results that they like, they go back and they combine the training group and the validation group and recut the original group and keep doing analyses. And if you do that long enough, it's sort of like multiple hypothesis testing, you can eventually find something that it works. These authors didn't do that. They made a hypothesis, they locked it in stone, and they analyzed their blinded validation set just like this. This article discusses chance as a threat to validity and a larger discussion of rules of evidence for cancer molecular marker research. I want to tell you a secret about this article, though. It has no new ideas. This is clinical epidemiology and biostatistics 101, just applied to an interdisciplinary field involving biochemistry and molecular biology. Let's turn to bias, illustrating with two proteomics examples. Bias is defined as the systematic difference between compared groups so that the comparison is erroneous, the result is wrong. And you'll get the idea, if you don't know this already from the illustrations. Bias is serious in observational or non-experimental research. Even one bias can be fatal, and here's some examples. Strong claims have been made that serum proteomics can diagnose cancer. Claims for multiple cancers of the ovary, prostate, and breast that reported high sensitivity and specificity, with results appearing in Lancet Clinical Chemistry, Wall Street Journal, NBC, PBS, Red Book. How many people here read Red Book? I usually catch a few people. I read it when I get my hair cut. I sort of want to know what people are writing. There was an article in Red Book that said, if you're a responsible woman, you'll have your blood drawn to check for ovarian cancer. And all of this interest led to plans for a commercial test, Ovacheck, in 2003 for ovarian cancer, but plans were delayed by the FDA. But the results that I'm going to show you and other results led researchers and funding agencies, if you'll pardon the expression, to redirect effort and grant proposals to follow these results. And here is the study, a study that set off very high expectations, use of proteomic patterns in serum to identify ovarian cancer, but there's other studies with similar problems of chance and bias. This is not the problem of one lab, but an entire field, and fields, plural, making weak bricks that if used in building can lead to problems. To be sure, the field is promising, but for us to explore it sensibly as researchers and as funding agencies, we must understand what we do and don't know, and we must not fool ourselves, Ed Feynman would tell us. The purpose of the study was to diagnose ovarian cancer versus not. The method was to take cancers and people without cancer, assess serum by mass spectroscopy. This is surface-enhanced laser desorption ionization. Was sort of the separation, and then there's a time-of-flight tube. Spectra are analyzed by a genetic algorithm. This is the sort of multivariable analysis that's done. For those of you who don't know what mass spec is, this is it. There's some proteins or peptides or something here that's going to be an ion source, and in this example you shine a laser on this and you separate these things, and then once separated they drift down a time-of-flight tube that's charged, and so the drifting is related to mass and charge, and things get picked up on a detector, producing patterns like this, peaks related to MZ values that have this sort of range, and these are the multiple variables that you can then analyze in a variety of ways to try to figure out, can I tell cancer versus not, or heart disease versus not, or people with a good prognosis and a bad prognosis for whatever it is. And the results were that the discriminatory pattern correctly identified all 50 ovarian cases for a sensitivity of 100% and a specificity of 95%. This is like saying we got the GPS coordinates on the Holy Grail. Just, call up motor pool, send out the Hummers, go pick it up. This is a really big deal. And it was in the Lancet, and it got a lot of attention from the public and from researchers who wanted to do this kind of work themselves, as you can understand. But this bias explains some serum proteomics results for ovarian cancer, as described by Keith Baggerty, a statistician at MD Anderson in Nature News and elsewhere. Was bias introduced by the run order of specimens, and here's what Keith figured out happened. If cancers and non-cancers are run on different days, and they were, Keith figured this out by looking at the raw data, that, to the author's credit, they published on the web, and looking at the chip serial numbers and so forth. And if the mass spec machine drifts over time, which we know that it does, then non-biologic signal associated with cancer versus not, it gets hardwired into the results because the things were run on different days and the machine drifts. That's bias. Bias or signal, it's there. Bias isn't detected or removed by splitting the sample into training and validation groups, what I showed you before by chance. There's really signal there, and it's getting picked up. For this kind of work, Keith Baggerty has been called the first forensic statistician, a term first appearing in Nature Medicine, so we know it's right. A forensic statistician is someone who tries to figure out, after the research is reported, what was actually done in the research. Keith deserves this title. It's fun. He's done great work. And by the way, Keith is speaking at the NCI this Wednesday, 2 days from now, at 11 a.m. at the Cancer Prevention and Control Colloquium at Executive Plaza North. It's not going to be webcast, so if you want to hear Keith, keep that in mind. But there's a lesson here. Keith's done wonderful stuff, but if we need forensic statisticians, it means that our journals are failing us. Our methods, results, and discussion sections are failing us. Remember what Feynman said? If anything could possibly be wrong, you must explain it. If we have to rely on forensic statisticians, we're not trying to explain it, we're not looking for it, it's off our radars, and it's off the radars of authors, reviewers, and editors. This is a big, serious problem. What is reported in published papers is so incomplete that if I want to learn the details of a study's methods, I go to a Gordon conference and take the guy for a walk in the woods and interview them like taking a history from a patient. How did you start? What happened next? What happened after that? It works. I have really done this. But the point is that we shouldn't have to be interviewing investigators to learn what goes on in a study. That's what methods and results in journals are supposed to do, but they're not. Here's a recent example where a different bias may explain discrimination. From the Journal of Clinical Investigation, it's entitled Differential Exoprotease Activities Confer Tumor-Specific Peptidome Patterns. And what this means is that patterns of peptides, little molecules, not proteins, is where signal or discrimination is. So digestion by exoproteases outside the body in the tube after the blood is collected, this is an important process to understand and to manage. But can bias explain discrimination? The promise is that the peptide pattern in serum discriminates prostate cancer versus control, and it's 100% sensitive and specific. And the interpretation is strong. Exoprotease activity should be the focus of future peptide biomarker discovery efforts, and low molecular weight biomarker pipeline is surging with potential, but might bias explain discrimination if the compared groups are different? The cancers were 67-year-old men, because, it was prostate cancer. The controls were 35 years old, and 58% were women. And this was in the JCI. Now, this difference does not necessarily explain discrimination. It really doesn't necessarily explain discrimination, but it's high in the differential diagnosis. Now, this may look in retrospect like an obvious and dramatic problem, but it received virtually no attention from the authors, reviewers, and editors in the original article. This kind of problem is arguably so important that it should have received extensive discussion in the text and possibly have precluded publication of such a strong conclusion. This problem may look obvious in retrospect, but when I sent this article and the supplemental material to the 30 junior faculty and fellows in the K-30 faculty development program that I direct at UNC and asked, can you find the bias? One person got it. And I asked her, how long did it take you? She said 4 hours. And this was a fully trained clinical epidemiologist who'd been through a 2-year program. In contrast, when I sent it to Barry Kramer, Barry was back almost within minutes and with appropriate chuckles. The point here is that asking what could be wrong and figuring it out in this kind of translational research can be very, very hard to figure out. Further, this kind of difference also suggests there may be other differences. Perhaps bloods were collected in different locations or clinics, different conditions that for a proteomics study could have a very big effect. Bias is the challenge in observational or non-experimental research. Bias is not icing on the cake. It is the cake. Bias is a large topic and difficult. There are multiple biases requiring different methods to address them, like randomization, blinding, uniform handling. Many of you are familiar with these things. But some methods aren't available at all in observational research. We can't randomize people to whether they have cancer or not and keep everything else equal. Some biases may be impossible to identify. Boy, that's tough. This is sort of like the unknown unknowns that Rumsfeld tells us about. In case control studies, there's biases that we don't even know what they are, so we can't even speculate whether they might have happened. This plagued the field of case control methods for years, trying to figure this stuff out. And in the face of all this, even one bias can be fatal. The process to deal with bias is routinely ignored by authors, reviewers, and editors in omics research, and I would dare say in other kinds of molecular and translational research as well. Bias is so serious that the results of a study are considered guilty of bias until proven innocent. It is un-American, but this is our approach. Innocence is proven by doing process. In design, you avoid bias. For example, in a clinical trial, we randomize people and do lots of other things. It's design. In conduct, you measure to see if bias occurred. In a clinical trial, for example, you ask people after the thing is done, did you break the blind? An interpretation. You determine whether problems, if they occurred, were they important, and then by reporting the process, this is important in methods, results, and discussion. You do Feynman. If anything might be wrong, you explain this. We do this in clinical trials all the time. It's expected. We know how to do it. We don't do it in other kinds of research. Let's look at how the process to deal with the bias of baseline inequality is handled in a clinical trial. Since baseline inequality could explain different results in the compared groups, you report the results of randomization to show, in Table 1, to show that randomization worked and equalized features like, well, in this study, Table 1 from the New England Journal, we're comparing one therapy to another, and we're looking at demographics and nodal status and histologic type and estrogen receptor status and so on and so on. There were no differences, and there almost never are any differences because it's randomized. But the lesson is this. In a clinical trial, you don't expect any baseline inequality because randomization, the method to equalize groups, is so powerful, and it almost always works. But there's a lesson here. The need to avoid bias and show that you avoided it is so fundamental that every clinical trial reports, usually in Table 1, the results of randomization. A whole page of real estate in the New England Journal just showing, hey, randomization worked. Okay, so Table 1 is required in every clinical trial to show that there's no baseline inequality. In contrast, the process to deal with the bias of baseline inequality in a typical omics study involves this kind of text. Let's see, there's some people here with breast cancer, obtained was not, age wasn't available, and so on. Let's go down here. All samples were stored at -80 until use. And that sounds reassuring. All samples were stored at -80. Well, that's good. But were specimens handled equally in all steps to avoid bias before specimens got received by your lab? Like the time from blood drawing to spinning and freezing, or the number of thaw-freeze cycles, or the duration of storage of one group or another, or the type of blood collection tube, and so forth. Any of these steps is a possible source of fatal bias in a proteomics study, signal hardwired into your data that can't be removed by subsequent analysis. It can't be removed by annotation or curation or multivariable analysis or principal components analysis or anything. It's there. Nearly every marker study is silent or at best very incomplete about these kinds of details. What might be wrong with this brick? We can't tell. It's not described in methods or results. It's not discussed by the authors in the discussion section. We may need to get a forensic statistician to find out. It's not good for the process of our science if we routinely have to rely on forensic statisticians. This slide shows how there are many different sources of bias that can explain discrimination in studies of markers and in other observational research. The intended comparison here is people with cancer versus without, and the red line shows when specimens are received in your lab, because you're the basic scientist here, this is the NIH, for you to discover patterns or analytes that discriminate. Things happen that can cause bias before specimens reach your lab to the left side and after on the right. In other words, there can be other differences besides cancer between the compared groups. For example, after specimens are received in your lab, differences may occur in handling, like the time when specimens are run, as happened in the ovarian cancer study, where cancers were run on one day and controls on a different day. Or differences can occur before they're received in your lab. Differences in demographics or method of specimen collection and so forth. Bias happened here to the left side when, in the prostate cancer study, when cancers were 67-year-old men and the comparison group was younger and mainly women. If you're the lab person, the biases that can occur on the right side of the line, they're controllable by you. They're in your airspace. But what can happen on the left side that can fatally compromise some translational research? You may not know what happened to specimens before they reached your lab, or you may ask and not be able to find out because those records weren't kept. And if you're a lab researcher used to the experimental method and not clinical research methods, you may not even know what to look for in this messy domain of study design and clinical epidemiology and observational epidemiology. For example, you may think you're studying RNA expression in cancers versus controls, but the cancers came from patients having surgery in the OR under anesthesia, under general anesthesia, while controls had needle biopsy in the clinic. That might be a what could be go wrong. to explain different results besides cancer versus not. Or you may think you're studying serum proteomics and cancers versus controls, but the cancer specimens were thawed and refrozen 5 times because other researchers found them interesting and asked you to take them out of your freezer, while the controls were never thawed at all. That might explain results. You can see from these examples that the kinds of bias that can occur are idiosyncratic to the technology that you're using and the biology of the problem that you're looking at, which is why you have to ask a general question and understand principles. What could go wrong? There are guidelines that can help us. There's Miami, there's Remark, there's MyAPI for proteomic studies. There's all kinds of guidelines, but they tell us what kinds of things to report, not what to do. And they also can't tell us at all the kinds of detail that we need to report. This requires depth, it requires understanding principles. Because all of this important detail, any important detail, can seriously affect the science. We need insight from scientists to do this, not checklists. Anyway, this slide showing sources of bias can now tell us the kinds of translational research most subject to the problems that I've been describing. It's observational or non-experimental research. And we're coming down to the last several slides. In contrast, if your lab uses the design of an experiment, biases may be avoided. If in your study you can select participants, people, nematodes, cell lines, they're more compliant, and randomize them to groups that at baseline are equal because of randomization, and then administer something A to one group and B to the other, and then assess the outcome or effect. If you use an experiment as your method, its strength, its beauty, is that bias may be largely avoided by randomization and other methods like blinding and uniform handling and so on. The problems of bias are there, but they can be managed, and that's why we like experiments and clinical trials so much and use them to answer questions whenever they can, but we can't use the experimental method for every question. And some lab researchers, maybe even some in this room, who are starting to do translational research are becoming observational epidemiologists, even if they don't know that and even if they don't want to. Don't tell them that it might be happening. We can't use experiments to answer every question. There's broader lessons for translational research from this illustrative example, molecular markers. If we ask what kinds of translational research do problems in that example apply to, is it just omics research, or discovery-based research, or research about diagnosis or prognosis in people or something else? The answer is this. Whenever the study design or method is observational, not experimental, that's when the problems we've discussed are most important. When the design looks like this, and when treacherous and potentially fatal things can happen to the left of the red line, that's when problems happen. These problems are discussed in this essay. Bias as a threat to validity. The title says of cancer molecular marker research, but the lessons apply to other kinds of observational and translational research. By the way, this piece also has no new ideas. This is Clinical Epi and Biostatistics 101. Let's return to Forscher, Bricks, and a conclusion. You remember the topic of bricks was only one topic discussed by Forscher, and it wasn't as important to him as building edifices. I focused on bricks because without strong bricks, we can't hope to begin to build a strong edifice. If bricks were faulty, the edifice would crumble. Weak bricks, faulty bricks, is a critically serious current problem, and it's one that we can fix, and we need to fix it. But Forscher's main point was edifices. And he also discussed really interesting things that you ought to read about in his essay. It's just 750 words, but there's so much there. Who should be a builder and who should be a brickmaker? And what about training? Are we training people wrong? Forscher beats up on training. The problem happened because a whole generation of people got trained the wrong way. Read it and see what you think. Lessons for translational research in 2007 are that faulty bricks exist in many areas of current translational research. Rules of evidence can help us test the strength of bricks and ultimately to build stronger edifices, and future efforts should emphasize faulty brick prevention. For the future, if we had time, and we don't, we could talk about a lot of things about how to fix this problem. What's the role of journals in being mediators for strength of bricks? What's the role of specimens and high-quality specimens for helping us to do exploration while avoiding lots of problems of bias? What's the conceptual framework that we ought to use to approach translational research? Can we rely just on discovery? Do we have to think about development, which is always sort of a second-class citizen? What's the role here? What's the right way to think about the whole process? Maybe there's something from drug development that we could learn that would be useful. Do we have incentives placed in the right way? There's attitude issues in there. There's lots of things in the et cetera that we don't have time to discuss. A few of the topics are discussed in this article about how to improve the reliability, that's the strength of bricks, and efficiency, that's a little bit about edifices, of research about molecular markers. Again, it says markers, but it's got lessons for other translational fields. This article is released in a digital form on the website of the Early Detection Research Network, or you can send an email to me, and I will be happy to send you electrons. You can find me at the UNC website. By the way, in this article, I think there are some new ideas. There's some, although much of it is still pretty basic clinical epidemiology and research design. Finally, as we look to the future, over time, we're going to learn strategies for doing translational research better, strategies that give us more reliable results, more efficient use of our resources. And as we do this, and as we think about this, and as we develop this, we need to keep in mind the fundamental principles of doing science and evaluating evidence. Thank you very much.
So just a note before we move on to the discussion period, and I hope you'll stay for a lively discussion because I found that very provocative. I hope you also recognize, because we provided the hard data, why we invited David to give this kickoff symposium, because I thought that was very remarkable. And we hope to continue in the same vein with different talks. The next one, we're trying to nail down some of the details, but it will be, again, challenge of common wisdom. It will be applied to emergency response to pandemics or nationwide emergencies, and I think that you'll enjoy that equally. So with that, let me turn the podium over. I think we have another 25 minutes, if you wish, to talk to David, and after that, if it extends, you can go out into the foyer with refreshments and ask him questions.
David, that was really remarkable. If you go to your final slide where you suggest the future solutions, I suspect it's not an accident that you've got the role of journals listed first.
Yeah.
And so I guess my question is, in light of the recent activity with the International Committee of Journal Editors and some of the guidelines that are now being imposed upon clinical trial reports, can you not, is it naive to look at that as a bottleneck or filter through which all changes or any changes that you might think would effectively reform the system can be imposed? Obviously, you'd need a lot of power there, but is it not the case that, and you suggested this with your sort of wry remarks about the New England Journal, that the big journals have a lot invested in their prestige as big journals, which presumably was earned because of their rigor. So why isn't that why isn't number one even in bigger print?
I think number one probably is the key. And your question is an important one about what lessons can we learn from our experience with clinical trials. I think as we step back and look at the big picture, we have to realize that the clinical trials are conceptually easy to do. They're experiments, and that's why they're beautiful. We've got now 40 plus years of experience with doing them well. We know what kinds of things to require. We know about how reporting ought to be done and so forth. And I think that there are several problems where we can't generalize to the field of molecular markers and in particular observational epidemiology except for the one thing we can generalize to, which is it's going to take a long time to develop. I think we still don't know what the rules are that we want to apply to observational epidemiology. We've got some idea, because this is what an epidemiologist talks about all the time, but they can't be codified and standardized, and we don't know them as well as we do for clinical trials. So if we don't know exactly what to do, it's sort of hard to set up the institutional arrangements like with journals to implement them, although this is a long-term goal. I think we need to figure out better what those rules are. It's not as if we have no idea, but they're simply not as nearly well done as for clinical trials. I've been trying to think, people pester me all the time, David, show us what does the table 1 look like for we don't see this in observational epidemiology. It isn't really done that way. The concept is there, but we don't handle things. Maybe we've got to get observational epidemiologists to move more in that direction. The short answer to your question is we need to learn, we need to think about what lessons can we learn from the field of clinical trials and how they're handled by journals, but I think we're decades away from that. But I think we could move much more strongly in that direction. Will you buy that, or is that too wimpy of an answer, or what do you think? I mean, this is a big swampy field. It's been underdeveloped for decades, whereas for clinical trials, you know, we know how to do them. We require, for example, with clinical trials, that results, we have registries now. That was one of the things you were referring to that journals have required. We have registries for all clinical trials, so we know how a body of knowledge develops. There's nothing parallel to that in basic science. And some journals even say, if you've got a negative result, we don't want you to send it to us. We want novel things. I mean, the whole concept of how knowledge evolves when it's anything besides a clinical trial, we don't have good structures to do that, and we haven't learned lessons from clinical trials. Susan?
I mean, the whole discussion related to journals is still retrospective. The study's already been done.
Right.
Okay? And given how hard it is to do this kind of work and the amount of resources that go into it, the time to really refine your question answering is before you actually do the study. So really, I guess one of the questions, I'd like to hear your thoughts in terms of how can we be more rigorous up front? And I say this as somebody who's constantly monitoring clinical and observational studies, and you're sitting there saying, oh, we shouldn't have done that in the first place. I know. How can we do a better job at that? Because it threatens the entire field.
It does. And I'll tell you how I interpret your question. I think you're saying, how can we ask good questions? And I don't know how to answer that in a short way. This is sort of like clinical judgment and what makes a good clinician. This is what makes a good scientist. Asking the right question gets you halfway to the answer. I can't tell you in an easy way how to do it, but it's absolutely critical. If we ask the wrong I think many questions in omics fields, they're really the wrong questions, and we're spending a lot of money doing the wrong things. That's something that in retrospect I think we'll be able to say we could have done a better job. How exactly to do that, I don't know, but this is the NIH, where the most sophisticated questions get asked and formulated and cultivated from people and so forth. I'm sort of teasing a little bit, but this is the heart of science, asking a good question, and I think you're absolutely right. We need to focus on that because we're asking often very mundane questions or off-target questions. I wish I could give you a better answer, but this is what makes a good scientist and a not good scientist, and it's critical.
You made a provocative statement that a whole generation has been trained wrong. So I would just like to provoke you into a few more details about that, and how could we do better for the current and the next generations? So
the questions about training, it wasn't me who made that provocative comment. It was Forscher back in 1963, and he didn't elaborate. This is a 750-word essay, and it was one sentence. He said, a great misunderstanding, this is sort of like the Bible, a great misunderstanding arose, and you try to figure out what does this mean. He said, and then it was in parentheses, some people think that it was, that this next generation of trainees was funded wrong, was trained wrong. He doesn't elaborate on how, but what he does blame is he said so much money became available to do research that people became obsessed with making bricks and getting funding to make bricks. And you look at what's happened to public funding in the U.S., lots of benefits, but Forscher's saying this was the cause of problems in the early 60s. He doesn't say any more than that, and I can't speculate on exactly what he was thinking, but he speculates that the problem happened when people got the wrong orientation. I'm not going to be a builder. I'm not going to try to deal with theories. I'm simply going to make bricks and generate units of knowledge. He said he's just saying people became oriented toward doing that, and they got rewarded with funding for doing that and lost sight of making edifices. I can't take it any further than that, but I think there's something important there. It's going to be hard to adjust, but, you know, there's something important. And it gets to Susan's question about are we getting people to ask the right kinds of questions, or is it questions that, you know, they can get funded for, and are study sections doing the jobs and funding imaginative things, and, you know, all the things that's the stuff that people talk about. That's what I think Forscher was raising. Yes, ma'am.
Yeah, I'd like to follow up on that, because you had talked about there was a clash of two cultures, and maybe epidemiology has some lessons for how to deal with the types of biases that are arising. But to go a step forward and to look at the culture of science itself and what could lead to these different things, because, you know, where you talk about the incentives have begun to change, that it's no longer good enough to come up with different research, but you have to be able to prove an outcome or to promote that, you know, while you're the basic of basic research and we haven't done enough repetition, what's the outcome going to be? That you're going to cure somebody from cancer, or to promote it back into translational, that a product's going to come of it. So there's these different types of things that are happening that are analogous to research integrity. What do we do within the community that there's enough time to train properly on bias and the inherent things in our culture that are promoting people to overlook that rigor and the time that it takes sometimes to do the investigation in the right way? Yeah.
How do we get people to be more rigorous? I can't give you a short answer, but some of the ideas that I've been fooling with or thinking about and would be interested in what others think, I think there's not enough negative feedback in the system when people publish something that may be wrong, in part because journals don't publish negative results. It can be very hard to even get a journal to publish a rebuttal to some article that got published. And so there's not that much penalty for producing something that's weak, because nobody ever knows that it's weak. I can't remember the phrase he used, but John Ioannidis, who's written some wonderful stuff in this area, he says, claims get made and you can't ever tell if they're right or wrong. For example, this claim about ovarian cancer and serum proteomics, this is 5 years after it got published, and we don't know whether it's right or wrong. People don't believe that it's right, but it's not as if we can point to clear, direct tests in the literature that say that it was right or wrong. And John Ioannidis has a concept that's sort of like the living dead or something like that. It's a claim, it exists, and we never know if it's right or wrong, and there's not negative feedback. Think in the old days, and it's very, very different, but it's fun to look at history of science, like at the time when people were trying to figure out the structure of DNA. And, you know, and you read the double helix and the biography of Francis Crick and Rosalind. It's thrilling stuff. Week by week, people would make claims and they could be right or wrong. If Linus Pauling says it's an alpha helix or whatever he says, and Watson and Crick say it's a double helix, somebody's going to know in a few weeks or months that a person was right and a person was wrong. And the way that things evolve now, there's claims and they don't get challenged, and we can't tell if they're right or wrong, and there's just not that much penalty. Penalty's a little bit strong, but, you know, if we want people to be rigorous, they've got to see that there's a problem with being not rigorous, and that's not happening enough. Yes, sir, and then here.
So I think, well, first, it was a really good talk. I think you put your finger on an important problem, and I was sitting here thinking about how 10 years ago I heard Chip Petricoin talk about some of these problems, and I was excited that someone was thinking about it in terms of that you would think a clinical researcher would, as opposed to an analytical chemist, that he was thinking a little more broadly about what needed to be dealt with and the observational part of the problem. I've also heard Paul Temps talk forcefully about the problems with the analytical side of it. And, you know, he's a good scientist, and yet these folks step in these chuck holes, and we all do.
They're becoming epidemiologists.
And if we go to you go to the mass spec meeting, which has grown to about 4 times the size it used to be, and you talk to the people who are presenting proteomics research, there's a sort of an unwritten or an unspoken attitude that a lot of what's presented is of questionable value. We know it, but like you said, there's no penalty
Yeah.
associated with it, and we don't, even though we know it, it doesn't stop. So I don't know what else you could say about it, but I'd be eager to hear it.
Well, you know, you say that we know it, but we don't. We don't know that we know or don't know something until it's been directly assessed in a scientific article. And this is what, you know, when Letterberg was saying here's 9 things that are subject to, you know, refutation and stuff, we just don't do that in a systematic way. So we sort of think that, gee, that's not valid, that's not real. But part of the problem is we're not sinking our teeth into stuff by having direct attempts at repetition or direct rebuttal. We're relying too much on forensic statistics to look at the inside. I don't know how to solve this, but I think it's a problem we've got to, as a field, to talk about more. Dr. Ferguson.
Yeah. I would like to know your comments, and then since we have a number of media here, their comments on what the media's role is in exposing these problems.
Well, I think if any people from media or anybody wants to say some things, that's, you know, that would be fine. I think the media have a role in this, but in my view, the media in general, I would hope that we as scientists would do the main bulk of the job. That's what we're supposed to do. That's what the back and forth is supposed to be, by trying repetition or looking at insights like Keith Baggerty and so forth. And to me, the main place that this needs to be worked out is the scientific literature. The media is sort of, it's not icing on the cake, but it's a little bit separate. I think the larger issue you raised, though, is that, you know, that attention and seriousness ought to be put on this. Barry, you run a thing for the media, and I don't know if you've got comments about this or if others do, but, you know, the issue is where and how does the scrutiny happen?
Yeah, we do, through the Office of Medical Applications of Research, have an annual Medicine and Media Symposium. It's designed to do some of these very things because, at best, I think sometimes the scientific community, at least in the current state of affairs, when they put out their product, may not even be aware themselves. They may be over-enthusiastic. They may be pushing a particular result and may put a spin on it. As a matter of fact, many discussion papers will bring up straw man arguments about potential weaknesses and then immediately state why those almost certainly don't apply in their own study. So at the next step, of course, the final conduit is the media, and we do feel that they are honest brokers of information. They're supposed to, at best, put things in context and translate them crisply, clearly for the public, and recognize that every study design has its strengths and limitations. Well, not every study design has its strengths. Many study designs have their strengths. All study designs have their limitations, and they should be transmitted clearly to the public. The problem with journalism today is training. That is, they're trained skeptics, but they're not trained scientists. And given the explosive knowledge in science, it seems like almost anything could be true. And so it's the wonder of science that gets reported. And the intent of our annual symposium is to at least give them the basic tools to read the article, and then question some of the problems about study design. And we're very proud of that seminar. But I agree with David that a lot of work has to be done on the front end. That is among the scientists themselves.
Yeah, I want to say a little bit more about scientists and our role. I'm a clinical epidemiologist. I'm a health services researcher who's gotten into the molecular field for a variety of reasons and worked with some specific assays and so forth. But I'm a clinician and a clinical researcher. But recently I've started going to some very high-powered basic science meetings run by the NIH and Gordon conferences and Keystone and the American Society of Mass Spectrometry and stuff like that. And to me, many of these meetings, the scientific papers aren't good. They're not nearly as good as the sort of fear of God kind of, meetings and approach that Alvin Feinstein taught us in clinical research. And I think the point is, I think the structure of our meetings is sometimes wrong when people present not 10-minute papers that's one fact that you sink your teeth into it, but a 20 or 30-minute thing from 30,000 feet with 900 facts in it, and nobody can sink their teeth into it at the end, and you simply don't know what's strong and what's weak. And to me, it's gotten to be boring and frustrating. I'm exaggerating a little bit, but not that much. Similarly, we don't do a good job with articles. I review for, things like Nature and Science, and, get articles from places that I didn't used to normally get. And in the discussion sections, typically what gets discussed is, if these findings are true, this is what the implications would be. What's missing is what might be wrong with what I just told you. If I saw a good faith effort to describe limitations like Joshua Lederberg, I would drop over dead from shock. Alvin Feinstein, when he was teaching us clinical research methods, said, and I remember this really well because it was very meaningful to me, he said, you know what, our field is second-class citizens because it's clinical science and it's so messy, and the way we're going to clean this up is by learning from our basic science colleagues. And Alvin Feinstein said he must have read Platt and Forscher, though I don't recall him mentioning them, but he said, when you read a discussion section in an article, if they don't tell you very early, these are all the things that could be wrong with what I just told you, Alvin said, don't bother reading the rest of the article. They're not scientists. I remember him saying that. And right now, we don't see this at meetings. We don't see people getting up and criticizing each other and sinking their teeth into stuff because it's too broad or people want to be nice. I thought basic science, I'm exaggerating here, but I thought this was supposed to be real hard-nosed stuff. Read the double helix. Read about the story of physics around the turn of the century and how the physicists battled each other in meetings. We don't see that now. I'm exaggerating a bit, but think about it, and if there's some lessons here, I think the point is that the media has some role, but I think we in the scientific fields need to do the Feynman and the Platt and the Joshua Lederberg and the people like that. Yes, sir.
Thank you for a very nice talk. I can't resist giving a little bit of a commercial, I guess, for clinicaltrials.gov, which does have over 50,000 clinical trials registered, with giving the design and giving you a chance to look at what's being done. Importantly, because of legislation that passed just several months ago, there will be a results database which will be available in about a year, coming on stream in about a year. This is important because about a third of all of the studies that are done are never fully published. And this may answer your negative results or inconclusive results. There's another side to it that many of us have thought about who are interested in clinical trials, and that is it puts on display all of the studies that have been done with all the warts and all of the difficulties that may have been encountered, and allows people to have their peers look at what they've done. And for many of us, we believe that it will change some of the behavior that is probably not as rigorous or robust as it should be.
Thank you for that comment. This is important, and I'm not sure how we reach it for areas where it's not clinical trials, but the basic concept you're raising is critically important. We're learning all the results in the field, and we're learning about negative results. If we have authors who don't want to publish negative results or don't even want to try to reproduce something because it's not sexy and it's not going to get published, we've got fields that evolve in crazy ways, lurching from one apparently novel thing to another without ever being tested. And the kind of approach that you're suggesting is something that in the long run I think we need to aim for. Yes, ma'am.
Hi, so we are here at NIH, and NIH not only does research, but it funds research, right? And as over the last couple of years, I'm in peer review, and the focus has been more and more on significance and approach. And we've been, as SRAs, we've been told that we should tell the reviewers that discussing methodology is not important.
Does it work?
Well, because of the shortening of everything, discussion period, it has to work. Yeah. And more emphasis has been given on significance. So we are moving away from what should be important and what would get us to where we want to be. If we do not discuss methods, then there is no way to find out if what is being proposed and what is going to be done. I see. So you're saying methods getting short shrift, and that may lead to problems. That's important. A very important issue. That is important. I have no suggestion about how to deal with that, but significance is important, but how you address it is also important. That's right, because that used to be a very important point.
Any
in peer
comments
review.
from others here who were involved with the extramural process? Susan, any comments? Anybody else?
Well, there are other people from peer review here. No comment?
Yes, ma'am. Oh, any other comments on this?
Okay.
Yes, ma'am.
I just want to make a comment, which is that it's methodology is a very difficult area to fund or to get funded in study sections. It's not considered sexy, cutting edge, discovery research, and yet everything that we do is underpinned by methods. But when's the last time we really had a major investment in looking at methodological approaches and new statistical approaches, A, and B, in educating all of our diverse scientific communities and keeping up to where the latest thing is in methodology. I mean, you're talking about training graduate students, and what about people after they get out of graduate school?
Are they trainable? I'm teasing.
It's a big problem.
Yeah.
And another thing, your talk was so excellent, and it also made me think about your comment that you are also a health services researcher and thinking about what is going on outside of here in terms of the focus at CMS, FDA, the Congress on quote-unquote evidence-based research, the push towards using more observational studies, saying clinical trials take too long or too costly, et cetera, and that we should be doing much more quick and dirty types of research, and where methods truly seem to fly out the window in many of the studies that are published that are observationally based. These are all concerns that I think you raise that are important to grapple with.
These are tough areas. One is you raised the question of how do we develop new methods, because that's not fundable. That is important. I think your point was, though, that just without developing new methods, we don't have time in study sections to talk about what the methods really are. And that's a separate set of issues. The last thing is this business. I don't want to trash all observational studies. That's most of what I do, because I'm a health services researcher and a clinician, and I don't do clinical trials. Observational research is not necessarily bad. This gets back some to Susan's question about how do you ask a good question? How do you ask a good question and think of a good method to answer it? A study doesn't have to be perfect, and that should not be the take-home message of what I'm telling you today. You do need to think about, and this requires clinical judgment, sort of in the realm of scientific research, is the problem that this is going to have when you think about it in advance. I think Susan or somebody said we've got to think about all the problems in advance, and we do. Is the problem that we're thinking about in advance, is it so bad that it's fatal that we shouldn't do it? But if there's nothing at all known about an area, you can't be expected to do a perfect study. It's got to be good enough and not have a fatal bias. Now, operationalizing that idea is another thing, but studies don't have to be perfect. And we necessarily have to rely on observational studies for many kinds of problems.
But I think the issues you raised here today really need to have more exposure and be on the radar screen. I think that's fine.
Amen. Yes, ma'am.
So I think I've been standing here long enough that my comment has changed about 6 times, which is not meant to say how long I was standing here, but how good the discussion is. But what I came up to the microphone to say, I'll stick to that instead of all the other things that have gone on through my mind since then. I come from a large-scale informatics background, particularly in genomic studies, and I think one of the real problems, in addition to lack of questioning the what could be wrong, or at least a contributor to that lack of questioning, is a divorce between the informatics scientists who are looking at the data that comes out of large-scale studies with the scientists who perform the studies, and with just a lack of appreciation for the science fundamentals. Computer scientists and mathematicians are not scientists, and I think that they don't question often enough.
Are there any of those in the room here? Are you guys going to take this line down? No, this is important. I think you're raising another area of culture clash, and it's between it's between information scientists and mathematicians, I think is what your point is. The generic, you can correct that if it's not. The generic issue here, though, is that people in many different fields need to understand each other's methodologies, but also what their purposes are, and what problems can arise when you try to answer questions using the other person's methods. It's a very, very hard and sophisticated kind of communication to get to happen. Is that addressing what you were raising?
Yeah, I think so. And one of the things I just say, that there's already the problem with scientists not questioning enough what could be wrong, but then to have another layer of interpreters looking at the data afterwards and either not being able to get back to the science question or just not thinking about asking those questions.
I find in the training program that I direct, and also at national meetings and talking with scientists and various collaborative things, they often take their data and pass it off to the biostatistics or informatics person and ask them to do magic on it. And Alvin Feinstein would teach us, you never do that. These are your precious data, and every data point you understand exactly where it came from. You can't give it to somebody else and let them just do stuff with it. You have to understand exactly what they're doing because it's so precious. And I see now, again, in my training program, but also the sense that I get at national meetings when people present their stuff, I gave it to somebody who needed a support vector machine, and here's some p-values and heat maps. And I think I could, if you asked them to explain in 4 sentences what those things meant, I don't think they'd have the slightest idea. It's a crazy disconnect, and I don't know what is going on here. I'm exaggerating a little bit, but not too much. Yes, ma'am.
I just add one other thing, that the scale of the data that are being interpreted right now contributes to that because the scientists aren't familiar with every data point anymore.
It does, but we can't, that's a problem. It's a solvable one, but we can't lay back and let high throughput stuff, there's still, common sense still has a role here. But I think we agree on that. Yes, sir.
Yeah, so I think really the question boils down to how good can an observational study ever be, because you will always have, hindsight will always be 20/20.
Right.
And some of the names on these studies, there were a large number of names on each of them, number one. There were maybe a dozen or 15 authors, and unless none of them paid attention, one presumes that these smart people missed some of those fatal flaws in advance. And so, and I'm no fan of Don Rumsfeld, but I mean, the term unknowns, I think it really
Some of these are known unknowns, though. That's the unknown unknowns we got problems with. If it's a known unknown, we ought to be able to deal with it. Go ahead.
But in terms of, concrete things that can be done, I think we probably have the best opportunity ever right now, because there's a very high-profile observational study, which was in the New England Journal, which is now getting the attention of Congress and the media like nothing, and I think it's going to get messier before it gets better. I'm talking about the observational lung cancer screening study.
I've heard about this.
Huge implications. Can you think of ways in using this opportunity to educate the media, Congress, and the public, and not least the scientific community, on how to design a good observational study?
Barry?
No. We can try. But that triggers a question that if we have time, I'll ask that. But that is a very tough nut to crack, because the very people that are engaged in the research sometimes are so taken with the dramatic results that they're unwilling to, it's easy to say in your heart's mind, I'm doing God's work, I'm trying to save lives. The results are so dramatic that it's hard for me to conceive that there are confounders that could possibly overwhelm the magnitude of the results that I think were there. And to me, I think that's just a lack of imagination rather than, rigor in the area of science. If the lower your expectations, the better your research looks. And the lower the amount of imagination you have to go through the list of 9 things that could have gone wrong, then the less likely you are to recognize the need for additional studies, or rigor. That's just my commentary.
Thank you. Yes. Yes, sir.
Yes. Great presentation. As a clinician, do you have any comments on the strengths and weaknesses of practice-based research, which seems to be becoming more and more popular?
I don't know that term in that field well enough.
This is a situation where you have networks of actual clinicians, practitioners, who are conducting research on problems that they feel are relevant to their day-to-day operations?
I'd have to look at the details. I wouldn't, off the top of my head, I wouldn't know of things that are inherently wrong, and things could be doable. But, you know, it's research that's subject to exactly the same kinds of problems. It sounds observational as opposed to like a clinical trial. I think you'd have to be careful about asking the right kinds of questions and anticipating the kinds of problems that could occur. I can't take it any further than that, and I don't have enough direct experience. Yes, sir.
I realize time's coming to a close, and I would love to, there's some, as wonderful as this discussion is, there's something about it of, I don't know, the emperor has no clothes, or you're preaching to the choir. Not that that's not a crucial message to deliver, but when I mentioned started off mentioning the ICMJE and a possible mechanism to do a correction, though, of course, your objection was apt. It's clinical trials and not epidemiology.
Yeah.
But in terms of epidemiology, I think that it's possible that one lever is this apocryphal cost of getting a drug through a clinical trial, $1 billion or something. And if NIH, as they are continuously under this budget crunch, markers, which was a significant element in your talk, can be a crucial part of epidemiological research, perhaps retrospective, perhaps prospective, but still, are you familiar with the Translational Research Working Group here has just sort of come out with a general structure about how to revise the approach to coordinating one step in the process from an idea to a final translation. And so my question is, would you think that it would be possibly useful to explore establishing at NIH, somewhat along the model that is being proposed, a sequence of funding steps whereby we control the asking and answering of some of these crucial questions, especially if you confine it to marker research as opposed to...
You're raising a really important, larger question, some of which is discussed in the article at the end in the Journal of Clinical Epidemiology that I noted. The question that you're raising is, do we need to see markers as an example in certain kinds of translational research? Do we need to think about it in terms of a development process like we do with drugs? Because we don't right now. There's what characterizes drugs is big investment. There's an agency, usually a drug company, that can fund something for 10 or 15 years or whatever, expects a lot of failure, and their job is to ruthlessly weed out failure at every step, in early stages or in late stages in drugs. For markers, for example, we've got no parallel process. What we've got right now is the equivalent of a lot of mom and pop labs that have one discovery that they hope is going to revolutionize the world, and they don't really want to learn if it's wrong. I'm exaggerating a little bit, but you get the idea. There's no grown-up, there's no journal, or there's no CEOs of a drug company saying, we're really going to put the screws on this and find out if it works or doesn't, because if it doesn't, we want to learn early. That's the sort of developmental process that there's nothing parallel to for markers that I think we need to think about. I'm not sure what the mechanism is and whether it's like the Translational Research Working Group or what it would be like, but I think I think we're going to in 5 or 10 years we're going to start thinking about the field of markers as an example of where development has to happen. It's not just independent discoveries by mom and pop groups, but there's got to be a process from going from one step to another and ruthlessly sorting out failure. Does that sort of address the concern that you raised? I think, in other words, I think we're thinking about things wrong right now. It's discovery and things are going to happen after that. That's not the way it works. We need to learn from our colleagues in the drug development business. And I think there's actually some easier steps that we can take, again, which are described in the JCE article. But our thinking along those lines, I think, needs to change.
It's 10 o'clock, and so we've come to the end of the discussion.
You're still in daylight.
11 o'clock.
Whatever it is.
No, I'm actually, I don't know what time zone. I'm in the Central Time Zone. We've come to the end of our session. I really appreciate you all coming. I know there are additional questions. Maybe you can pursue them when you join us for refreshments. Please do. Refreshments are out there waiting for you all. And thank you for coming to the inaugural session of our medicine conference.